Okay, problem sorted. It also I have more like plant taxonomist background. I've done mostly Sanger sequencing and a lot of DNA extraction. My colleague Lasha is a bit more proficient, much more proficient into the genomics world, into the new trends, into the computational part. So in a way we complement each other when I'm going to give a bit of a background within the field and lab parts, and then he can step in and talk about the newest cool genomic stuff and computational analysis. So we'll start by talking a little bit about DNA sampling, things to think about when you're using herbarium specimens or your collections for molecular work, an overview of the laboratorial techniques, and in the end, the phylogenetic analysis when Lars will join. So you can use for your studies anything from a leaf sample to herbarium specimens. You then take it to the lab to extract the DNA, you amplify the region that you want, and then you sequence. Some institutions do have their own sequencers, but it's usually very expensive. So most commonly these days, you do the extraction in the region, selection, amplification in your lab, and then you send the samples out to a company to generate the sequences. Then they are sent to your email, and then you can start the bioinformatics part. So this is how everything is connected. So when it comes to DNA sampling from the field, it's very common for people to carry silica when they go to the field, thinking already of collecting DNA samples. So usually carry a little bag with silica and some tags. So just if you want to be prepared, just make sure you always keep these separate bags. It's easier to collect the fresh leaf on the spot rather than pressing the specimen and then trying to collect the leaf later. The DNA will be far less degraded if you put it straight away into silica. So people do that along with collecting experience collections. You do the same thing at the same time. So very important when you're doing that, just keep a good record of your samples. So not just your field notes, but also a separate notebook with what you're collecting for DNA samples. And try not to mix them up. That's easier said than done. When you're in the field, you're rushing to press the specimens, to mount them correctly, to change the sheets. Keep your DNA samples always well organized. You're going to thank yourself later because you only have the leaf. So when you have a specimen, you have other elements that help you identify it. But when you only have a leaf in a bag and a number, it's so easy to swap. So just be always very careful with your samples. And always collect a voucher specimen. So think the other way around. If you're collecting a specimen, try to collect a separate DNA sample. But if you're out in the field to collect DNA samples, don't forget to also make a voucher specimen, even if it's just the one to put in your local herbarium. And I know some people forget about this and they go on and do a whole project, and someone asked, where is the specimen attached to these samples? That, oh, I forgot, I only collected the DNA sample. So this is very important, not just for DNA studies, phytochemical studies, anything that you collect a sample. If you don't have a lot of material, at least one plant needs to be pressed and sent to a herbarium so that if anyone wants to see what the plant looked like or you want to re-identify, you know it's there. This is important. So what's the advantage of collecting the DNA sample from the field straight away when you're out there? It's fresh, and we know DNA degrades over time, so the fresher the better. If you're going straight into the lab, it's great to have the leaf just straight from the field. You have more tissue available, so you have more leaves, stems even, to choose from. You've collected it yourself or with an expert, so it's more likely to be accurately identified. And contamination is also less likely because you've known what you've collected, you've been careful, it's your project, so you're a bit more in control of what's happening. The disadvantages is that if you're storing these little samples, you're going to need dedicated space in your institution, in your office, that takes up a lot of space. Also certain temperature and humidity conditions, so they can get moldy if you don't keep them correctly. So you have to think about whether it's worth keeping those samples or just using them and then throwing the rest away and just keeping the specimens. Also, you can't rely only on fresh material because it's a more recent practice, so you only have for a few taxa or for some regions. So only the field sample might not be enough. And also the silica these days is okay, but most of the silica available in many herbarium is still toxic and hazardous to handle. So you have to be careful not to inhale the fumes. You have to be careful where you dry it. Don't dry it in a kitchen oven where people are going to eat, and this kind of caution. So there are these days the silica is okay, but not many people have access to the modern types of silica that are not so toxic. So these are all things to think about before you think it's great. I'm only going to get samples from the field. Just some caution. But think about the fantastic resources you have in the herbarium. You have thousands, millions of specimens that you can use from a lot of locations. So if you're for your project, you don't need to actually do field work to these places unless you really want to. Some regions will be already represented in the collections. Lots of species. They're also easier to store in a way because they're compressed, so they're two-dimensional. It's easier than storing all of those silica dried samples. And there's also documentation of the species over time. So it's been collected in one location in the 1930s, then again the 1950s, and then you don't have any more collections. It can tell you many things. It can tell you the leaves are getting smaller. It can tell you that it doesn't occur anymore in that place and you need to go and check. So there's lots of reasons why you would want to sample across different times. So all of these advantages of sampling from herbarium specimens. You can see I'm in defense of using herbarium specimens from molecular studies. Sorry, but what are the problems? And we all know there are many challenges in using herbarium specimens. So DNA degrades over time. So we know the older the specimen is, the more fragmented it will be, and I'll explain a bit more about that later. It will be treated with chemicals, which is also very bad, but we know it's necessary in the tropics. Sometimes you have no choice, you have to use chemicals. Also in some institutions, the specimens are actually glued to the sheet, so you can't take with it.
Just sorry.
So you can read the okay. So sometimes the material is glued, so it's inaccessible. You can't always trust the identification, so either you have to re-identify yourself, just check it, or run it through experts that you trust. And also we have to be careful about contamination between samples. So one of the examples I have here is when you have a fragment packet at the bottom. So if you have here this fragment with lots of leaves, this is actually a paper that we published because colleagues unfortunately took a sample from this packet and they concluded that this species belonged in a different family, but actually there had been a mix-up in the herbarium mounting process where a leaf from something from a different family had been put together. And this is a very straightforward example of a contamination during processing of the material that you have to be aware of. That's why I said when you collect it in the field, you know what's happened to the material to that point. But when it goes for mounting, when it goes for processing in the herbarium, many things can happen. So when you look at a herbarium specimen, think about the history of that sheet in front of you and whether there could have been reasons to suspect that this is not what it says it is or there's some mix-up. So always be careful in that respect. So again, beware of cross-contaminations, so there could have been even mixed collections in the field, so sometimes you have more than one plant on the same sheet. It's very easy for similar species to be put on the same sheet and be assumed to be the same thing. Yes, species that look alike. Sometimes incomplete specimens are put together, so the inflorescence of one species and then the stem of another one and the root of another one are put together. So if you're sampling the leaves, you have to make sure that those leaves correspond to the rest that's on the sheet. And again, what I was saying about the identifications. Can you trust the identification? Are you sure that what you're collecting is exactly that species? Also be careful with nomenclature. If you're assuming that the specimen on the label is what it is, check if there's a synonym, check the authority, so be careful with species with the same name but a different author. Check all the different possibilities, because this can generate a mix-up in your specimen labeling. If you can, run it through an expert of that group, and also think critically. So if you go through all the process, you will have your results, and it's completely unexpected. Like in this case, well, it was unfortunate for our colleagues because putting the species in a very, very, very different family. But this is something all of us have to think about when we get our results back. Could it really be in that family? Could these species really be in that family? Could something have gone wrong? And that's not a problem. If you go through the whole process, you've got your sequences in your email, you analyze everything, and there's something wrong. Well, that's science and mistakes happen. It's more important that you stop and you go back through all of the steps back to find out what could have happened even at the level of the specimen itself, than to go forward with those results. So it's very important to know from the start where the mistakes could be, so that when you get the results, you know what you're thinking about. Okay, so this is the paper. If you did a bit of self-promotion, but it's been lots of fun to write, and a lot of people have been using it as a reference because we give a lot of recommendations of things to be aware of when you're sampling from herbarium specimens. Okay, so this is kind of the overview of how to use samples in the field of herbarium. I guess everyone kind of knew about this. Yeah. Not everything. New things to think about. Okay. Okay, so a reference that could be useful. I've always struggled while teaching future taxonomies about molecular techniques, because I've always thought that the references available were too complex. They're too oriented for geneticists, for molecular biologists, and it's very difficult for someone with a botany background to read through these books and understand, oh, which technique should I be using? What's the problem here? So we've actually got together, I was part of this exciting project where we wrote a book specifically for botanists, especially for plant taxonomists, where we break down all the techniques with the goal of using that data for taxonomy and systematics. So the chapter 1 that I was mostly involved in was the extraction part. Yeah, so that's the one I've been involved in. I'll just give an overview of the different parts of the lab methodology. Is that okay if I go through with that? Yeah? You want to stop, or is that okay? Good to go? Yeah. Okay. So the several steps of the lab work. So you take the samples, you extract the DNA, you copy the regions that you're interested in for your analysis, you send the material for sequencing. Normally it's outsourced, so you send somewhere by people. You get your sequences back, and then you analyze them on your computer. So for the DNA extraction, it usually starts with crushing the leaves. You can either do it in a pestle and mortar, or you can put them in little tubes, and there are now these mini pestle and mortar things that you can use inside the tube, so you don't have to use very big equipment. But so it's kind of called you're manually pressing the leaves to make them into powder, or you can also do mechanically. So if you have in your lab one of these automatic tissueizers, it's very, very handy. You might still need to prep them manually. It helps a lot. So you add a bit of sand or silica beads, and they all go away afterwards in the extraction process. So that's okay. You can keep the silica and the sand in. And then you just, if you have this machine, you can throw lots of samples in there. It goes.
Yes, we are having this.
You have this one. Oh, that's so much fun. So many labs don't have it, and it's so much hard work. In Brazil, we didn't have it. Oh, it was so much hard work, sample by sample, just manually. So that's great. Use it.
We are using it.
It's much faster.
Yeah.
Do you add something to it?
Yeah, the buffer, and also in some cases liquid nitrogen also we use.
Yes. Yeah. Liquid nitrogen as well. Yeah. Yes.
And buffer is also we are using when you are working with the kids.
With kids.
Then in that case the buffer solution we use. Otherwise in the liquid nitrogen we use on that.
Okay. And do you just use the kits, or do you have to do
Both.
Mixed?
Mixed.
That's what I've worked on as well. Okay. So once you cut your leaves, you get into the CTEP extraction protocol, which some labs have kits that do all of this for you. They have a buffer, and they have the salt, and they have everything you need. But you can also do it kind of manually, more old school, with the actual reagents. And it gives you more freedom. So the kits are great because everything is standardized. In some plant families, it just doesn't work. In Congo-Glacier, you can't use the buffers and all that part from the kits because we get no DNA out of it. There's just too many phenols in it. All sorts of secondary metabolites because many species are medicinal. So we have to tweak everything manually, so we can't use this part from the kits. But this is called the CTAB, normally people use the CTAB extraction protocol, and this is what you will see in the literature, and often you will see the CTAB modified protocol. So this is kind of, well, I found a figure that kind of summarizes it well. And one of the crucial steps, and again, this is from my experience as a taxonomist, I don't know if someone would have more background molecular biologists might discuss the other steps and how important they are, but this step of the organic phase extraction and whatever you use in it is very important because it's kind of family dependent or group dependent. So for each plant family, you might find you need to tweak it a little bit to make it work better. Either because there's some sort of chemicals used in the herbarium specimens, alcohol, sugar, latex, oil, medicinal compounds. So for that reason, if you use your kit, it doesn't work, and you need to rethink one of the steps because it's not working. This is the first step you should try. Something's not working, then it's got to be the salt you're using, it's got to be the amount of buffer. You might need to add something else to it. So read the literature of that plant group to find out what are they doing differently to make it work. And as I said, it's usually in that step. And when it comes to herbarium specimens, so I was just talking about specifically about working with old herbarium specimens. So this is from my perspective. The age of the specimen is not really a chemical problem. So when it comes to extraction, the extraction itself, it may not be a problem. It will be more fragmented, it will be in smaller pieces, but you still may get good quality, clean DNA out of your specimens. The issue is different, is that usually older specimens have been poisoned a lot, so more than the more recent ones that have been exposed to a lot of chemicals, and they are most likely also to have been collected in alcohol. It's not that these days people don't collect in alcohol, but if they want a DNA sample, they collect the whole plant in alcohol, but they keep a separate sample that has not been collected in alcohol because the alcohol brings a lot of problems into the extraction. And there are specific CTAB modified protocols prepared to deal with alcohol collected samples. So you'll find, I'm not discussing that here today, but there are protocols just for that. So this is mostly the issue with the old herbarium specimens. You have to find out whether it could have been collected in alcohol, how many chemicals it's been exposed to, or, honestly with the poisoning, I've kind of I've been very frustrated most of the time, end up giving up because if you try many times and it doesn't work, sometimes there's really, not that I know of at the moment, of any protocol that deals specifically with this, and we just end up just not using those samples. But with alcohol, it's still possible. That's okay. We've found ways around it. So that's mostly the problem with the old herbarium specimens. If it's been well collected without alcohol, dried in the sun, it has not been subject to any poison, it's fine. It could be 200 years old, but chemically it will be fine, to some extent. At least the extraction will be fine. Okay, so once you've got your DNA extracted, well done. Well done. Then you think about, what do I want from my DNA for my project? And you can store that extracted DNA for months and take out little bits of it for the different projects to do different things, depending on the region that you want. And that could be even entire genome, or it could be just small markers. So the first step, extract DNA, is one thing, and then what do you get out of it, which regions, is the second part. So you can either get selected genes or selected markers, or you can get these many genes at one sort of genomic scale amplification, or you can have the entire genome as it's being done these days. So this is a very simplistic way of putting it. This is again where my non-molecular biologist background comes in, but I hope this helps to organize in your heads. That if you're looking at a couple of markers for your study, you'll probably be doing Sanger sequencing. If you're generating hundreds of genes all at once, you'll be dealing with next generation sequencing, and then you can also think about whole genome. So Lars looking at me thinking, all good. So I start sweating now because I get into this molecular stuff. But okay, so this is kind of the three different methods in a very general way that you can use for your project, according to your resources, according to your question. So the Sanger sequencing and the next generation sequencing, they have kind of a similar structure in the way the methods work. So if I go quickly through the Sanger sequencing, you'll easily understand the next generation as well. So you need your primers, which are small sequences that are going to tell the enzyme which part of the DNA you got. So like little flags that it's going to go direct into the DNA and say, here, this is the part that I want to copy and not anything else. And usually there's a forward and a reverse, and it will be, yeah, you add the enzyme, sorry. You add the forward and the reverse sequences of your primers, the little flags. You add lots of bases, A, T, C, Gs there, and you add your enzyme. You put it into the thermocycler, and through a series of cycles, it's just going to keep copying that region over and over and over and over again. So if it's 30 cycles, it's going to do that 30 times. And there's going to be temperatures that could be specific to some regions, so you have to be careful with that. Look at the literature. What was the temperature of annealing? What is the extension time of that? But that's what you use a PCR for, to copy just that region. So in the end, in your little tubes at the end of the PCR, you will have a bit of your original DNA, which wasn't copied, well, it's still there. A lot of copies of only the region that you want, so that's going to be the majority, so the rest is kind of neglectable. It's still there, it's always going to be there when you send it out for sequencing, but that's fine. The majority is the region that you were interested in. And a bit of contaminations, errors of copying, and other things which are kind of minor. So you should know what is in your tube at the end. You know, don't only get the region that you want, but it will be kind of most of it, so that's fine. So then how do you know if it works? People do this in different ways. I usually just run a gel with allophoresis, and you see the bright bands will tell you which samples worked and which ones didn't, but it can also tell you if the DNA is very fragmented or not. We're not going to get into how, but just so you know, there's more than one thing you can get out of this gel. It's not just a yes or no, did it work or not. It can also tell you that the fragmentation is important, because if it's a herbarium specimen, you will see it already in the gel after DNA extraction, if it is very fragmented or not. And if there's any contamination, you usually get more than one band because it's more than one species. So all of this you can get from your gel. And you can also use something like call it a nano drop, other equipment. So I'm using a brand here. I'm not paying me to say nano drop. Oops, okay. But there's this quantifiers, DNA quantifiers, so they will give you a concentration of DNA, RNA, an approximation of the purity of DNA, and can give you information as well. If you have contaminants, what are they? So salts, protein can still be there. If you have phenols, I know in probabilistic you'll usually see a peak that tells us that the phenols we're trying to get rid of are still in there. So we can even see through that curve what else is in that sample that you couldn't get rid of during the cleanup. Okay, so that was the lab part of the Sanger sequencing. And now, running through another thing which people ask me a lot is, how do I know which markers to use for my project? Like, where do I start? Because I read this paper and they've used these markers, but in my lab we have these other markers. Should I really order those markers? Should I just try to do something genomics because it's cool and it's what everyone's doing? Maybe I should be doing Sanger because it's outdated. And we'll have all of these questions. So, very briefly, very simple way, try to go through that. To give you a starting point. So as you know, the DNA we usually work with is either the chloroplast or the nuclear. Of course, there's the mitochondrial as well, which we don't usually use in plants. The chloroplast DNA it has a maternal origin, so it's more conserved through generations. It has far less variation. And it's very suitable for higher level molecular phylogenetic analysis, so at family level, at tribal level, because it's more conserved. I don't know, that's making sense to everyone? Yeah. The nuclear DNA is far more variable because it suffers from crossing both parents, or mixture of the DNA of both parents. So it has more variation, has been more altered through the generations, but it's very useful for sort of lower level taxonomic studies, so at population level or closely related species or small genera. They are both important, but they can be used in different ways depending on the scale that you're working on. And then, of course, we get into population genetics. You might be looking at microsatellites, RFLPs. That's all nuclear data, but we kind of just go deeper into the level of variation. Then barcoding. It's another type of markers. The concept came from zoology. So they were the first to come up with this idea that you could have a unique sequence, a unique marker that you can sequence everything with. So these have been also introduced in plants. If you hear about barcoding markers, they're usually MATK or BCL. They're supposed to be universal markers that will work on any species. And ideally, if everyone sequences that, you can use it for any level taxonomic study. Well, kind of. Yes, exactly. In theory, but in practice, you find many groups where RBCL has zero variation, you don't do anything with it, and for other groups it's really informative. So I'm also kind of the opinion that this proposal doesn't really work in practice, but if you hear about it, you know what barcoding means and what's the intention behind it. Okay, then we also have different types of markers. So we have coding regions, so regions that actually are intended to translate into proteins, those are genes. And then you have these other regions in between which don't code anything, and we call them non-coding regions. So it's going to be spacers, peaks, anything like that. So you can hear genes, markers, spacers, and these names are very confusing, and we're not going to get into the details of the actual definition of each one of them. But in general, they're all markers, so they are regions of interest for our study, but some of them will code, and others are just garbage DNA kind of accumulating without having a particular function. And they're all important for phylogenetics, but in different ways. So the coding regions, they're important because they have a function, so they suffer selective pressure. They don't work, they go. If they work, they stay. So they are slower to evolve because plants don't just get rid of them, otherwise they can't do some of their metabolic pathways. So they have less variation because they're doing a job, so they have to be that exact sequence or else it doesn't work. So they're less variable. And because there are repair mechanisms for these genes, if there are mutations, they're just quickly repaired. So they're less likely to be variable in their sequence. And then the non-coding regions, they have just no selective pressure, they're living the good life, they're just accumulating mutations as they like, and no one's checking on them. So they're less conserved through generations, and they accumulate more variation. And mutations just happen randomly and are not corrected. So, for example, within the chloroplast DNA, you might choose to use some of these non-coding regions for more variation instead of the coding regions. Okay, so this doesn't work for everything, but it's something to get you thinking. What is the best strategy? And I always suggest a combination of characters. So if you're working at a family or tribal level, some sort of high level, choose slow evolving, more conserved genes so that they can give you a good structure at the high level. Otherwise the sequence is all going to be the same at species level because it's so conserved that they just aren't going to share the same sequence. But the higher you go, the more generations back, you're going to start finding the variation you need. So family or tribe level, possibly non-coding regions and some coding, but focusing more on chloroplast. At generic level, you need not so slow, but not so quick. So you might explore a combination between chloroplast or nuclear DNA, but check the variation within each marker, thinking, oh, I don't want something too variable. You want a nuclear marker that's very, very variable. Otherwise, it's going to give you random results. But you don't want too conserved, otherwise there's no variation at all. And at species level, you do want something fast evolving to give you enough variation between the species. So for those, usually the best candidates are nuclear DNA with one or two chloroplast DNA markers first. And if you have very, very close species, you have to think about, do you really want to do a molecular phylogeny, or do you want to more like get into population genetic study, or if you have populations that you're not sure there are one or two species, that kind of thing, you might then get into microsatellites and RNAs, right? So I hope that was not too confusing, but it's just to say there's not one solution for every problem. So some people say, oh, I'm working with these species, so I'm going to use this marker because I've seen it in the phylogeny of the family that they've used this marker. And I get the person to think, but wait a minute, they've done a family level study and they've needed to use this particular marker. You're going to work with a small group of species, they are very closely related. If you're going to use the same marker they used in the family phylogeny, you might not get any variation. So it's not just for that species use that marker, it has to do with the question you're dealing with. If you need to choose your own markers, if you can't find them in the literature, I suggest this paper where they give you a comparison of markers and they give you the programs, the PCR protocols, the temperatures, the primer sequences as well if you want to order them from the lab. So for my PhD, I had to choose a new combination of markers, and I started with this paper. So I chose a few very variable ones, a few more conserved, and I tried them out, and in the end selected a couple that worked better for our group. And then again, you can also check the specialized literature of your taxonomic group. Of course, it is good that you use the same markers and the ones that are already available, but be careful that your research question is not too different from the paper that you're using, just because it won't work. But if it's a similar question, like if it's also at genus level, at species level, then it's better to use something that's been used before. There's going to be more data available. And also for the future, it means your data is going to be integrated into other studies as well. Okay, so that was Sanger sequencing, which I think is our bread and butter, as they say, of taxonomists. I don't think we're going to go away completely from Sanger sequencing. It's cheap. It often gives us just enough for the kind of questions we're dealing with. Lars might disagree. Oh, he's quiet. Okay, he hasn't shouted at me yet for that one. So I sound old school in that, but I do think that if you got a pen and paper to write, then you should use a pen and paper to write. So if Sanger sequencing and a couple of chloroplast and nuclear markers are helping you with your research question, there's no reason for you to go to another step and generate lots of genomic data, get into lots of trouble if that data is giving you what you need. But you need to go step by step and start exploring the data first, right? So if you do use all of those fantastic markers, you try a different combination, coding, non-coding, chloroplast, nuclear, 3 chloroplasts and 1 nuclear, 3 nuclear and 1 chloroplast, and you're just going crazy, nothing's working, you might think, okay, maybe I need more data. And then you get into this genomics level of things, and you might start exploring the available opportunities out there. So what colleagues, some of them from Kew, have developed, but it's been a combination of people from different institutions. They've developed a kit that basically will fish out about 300 nuclear genes out of your DNA just at once. So that's basically what it does. And that's great because you get a lot of nuclear data. And then they're now using it for angiosperm level palynology. So kind of a new APG, a new classification of angiosperms. And so they're proving that nuclear data can be used at higher levels after all that I've said. Actually, our colleagues are using nuclear DNA for a higher research question, but it is a lot of data, so 300 nuclear genes is a lot of data. But it's the same concept as the Sanger sequencing. You have the primers, and you have the enzyme, and it gets copied, and so it's kind of the same thing, but it's just more data. And then you have other techniques that work in a very different way. So they're not just the same as this. The same concepts. So we have genome scheming, all sorts of type-seq techniques that I'm not going to get into because I wouldn't know. But when you get into the genomics, it's still developing, it's fast evolving. Every day there's going to be a new thing, but be careful when you get into it. Just make sure that you really cannot do it with what you were doing so far.
Now I have a question. Actually, for herbarium specimen, which method is better than the Sanger or the next generation? Because in Sanger case, actually the DNA is much more degraded from the herbarium specimen.
Yes.
So the standard markers usually in many times are not working there. So for that, what type of protocol we have to develop? What type of marker we have to select with the degraded fragments? Or we will go for the Sanger one? Sorry, next generation one.
Yeah, that's actually my next slide.
Okay.
And it's something I've experienced recently myself. When I've done a whole plate of next generation with herbarium specimens, and we had a lot of problems, but not with the extraction. So it extracted well, but then with the copying, we're not quite sure what happened. But at some point, everything failed; it just wouldn't copy, and it can't be too fragmented. So I think this is a key issue. It can't be too fragmented that it's smaller than the primers, because then you can't copy it. So you have to know the length of your primers when you're using your bait sets on next generation, and know if your fragments are long enough that they will attach to the primer. So in our case, that's the good thing about next generation sequencing, is that you have lots of control steps, and you can check exactly how big your fragments are, and if they're too long, you can fragment them to a particular size. So you have a lot of control of how long your fragments are at the end, which with Sanger you don't. You just get what you get and you try to sequence it. But with NGS, you can try to put into a certain size, but it only works if they're long and you're trying to shorten them. If you read them, you make a reading of the size of the fragments, and it tells you it's like 40 base pairs long, 100 base pairs long. That is so short, it probably won't even attach to the primers properly. And that means you can't amplify it. It means that the library preparation is going to fail. So this is what seems to be the problem with too much fragmented DNA. But I think at this point it's a lot of trial and error. I think people are trying things because it's true. Fragmented DNA that doesn't work with Sanger then can work with NGS, because you actually need the fragments to be smaller. Just as I said, not too small. Let me think. Something else I want to say about that. Yeah, I can just go through the comparison between the two now. Even though I can't give you a proper answer. You can just say I struggle with that too. So if you use Sanger, you can either get small or larger fragments of DNA. You have to do PCR. You use fewer genes. It's cheaper. The techniques are far less specialized, so usually undergrad students, MSc students can pick them up very quickly, and it's fast to go through techniques. And usually any personal computer or laptop can analyze the data very quickly. So that's the good thing about the Sanger data, that if it suits your project, that's great, because it's so much easier to work with it. And then the problem is that few data sometimes cannot provide good enough phylogenetic results. Sometimes you do need more markers. And actually what we just said could have been added to that slide as well, that sometimes if the fragments are too long, you don't have really a way to shred them and to make them smaller. So you don't have control of the size of the fragments. In next generation sequencing, well, and then again, sorry, and then if you have herbarium specimens where they are already small, they just won't amplify because you need very long sequences. The markers are much longer, can be much longer in Sanger. So in next generation, you get immediately much smaller fragments, and therefore with herbarium specimens, it just works better. Or you can fragment them to the right size. There's a range of techniques that you can use for amplification or to gather the data that you need, but it is still quite expensive, much more expensive than Sanger. Also, you need really knowledgeable lab technicians. And as a plant taxonomist, I've been arguing for this. It's not a very popular point of view, but I keep saying that I don't think we should be training plant taxonomists to do the lab work of the genomics, because it's so complex. And not everyone is trained in molecular biology to know what they're doing in the lab and to troubleshoot, because it's not just following the protocol. You run into problems. You have to think about all of these issues of what's going on inside the tube, what's going on inside the machine, so you can solve the problems. I defend that if possible, either you have one or two people very well trained in your lab who can help everyone else with the work, or you can hire someone just for that. We very recently at Q hired someone, and it's been a bless, because all of us having to do our own lab work when we don't even have students. I was having to stop my meetings to go to the lab and then come back. It's insane. And we realized you can do that with Sanger because it's so easy. In a couple of hours, you can prepare PCR, go away to the meeting. With genomics, you really need to stay on top of what's going on. It's very sensitive. It requires a lot of molecular knowledge. So I think it's better to have a few specialized people and plant taxonomists who understand the techniques enough so that they know if that's what they want for their project or not. So you have to know what's going on, what's happening with your samples, but not necessarily that you do it yourself. So that's kind of my point of view. And it also needs a lot of computer space. It might take weeks or months to analyze. It's computationally very complex, so you have all these tiny fragments, and it's like Lego. You have to put them all together, and you have to have a reference genome to help you assemble it. So it's very complex at also the informatics level. So some institutions are also hiring someone just to do the bioinformatics of the genome. So you need a lab person, you need a computer person, just for those specific tasks. But it does generate a lot more data, and for some very complicated taxonomic groups, it does generate better results phylogenies. So it could be worth investing in these techniques when nothing else works. It's very exciting. It opens a lot of avenues for different research questions, not just systematics, as you know, so transcriptomics and function and understanding what the genes are for, all of that. But yes, for phylogeny, it's great, but it also comes with all of this that we need to be aware of. So just the last one on whole genome. So we have to think about all of these. You can use the same DNA extraction methods, and it's just what you do later with it that changes. For whole genome, there's this fantastic new technology for which you don't need to assemble the fragments. It will give you the whole genome in a single read. So that's what we call the fourth generation of Pack Bio is the name of the company who does it. And it's great. You only need to extract the DNA, send it to the company, and you get the whole genome sent to you at a very high price. Well, not so high these days. I think Lars might know the budgets, but it is much more expensive than Sanger, for example. But you don't need all the bioinformatics to assemble it, for example. So it's much easier. It's getting less and less expensive, so it is a possibility. But even so, you have to think about for your project that different genes evolve at different rates. So if you're using much more data, think about which ones you're going to need and what's going on with each gene. So you can choose the best analytical methods. What are the computational possibilities? Do you have enough space to run this analysis? So before you start getting excited of putting together a project, and I've seen that happen, unfortunately, people get very excited, they generate the data, and they don't have the computational possibilities to handle that kind of data. It starts taking months and months and months, and then they need help from another institution. It gets complicated. So before you launch yourself into the lab part and start spending money on the sequencing, just find out if you're going to be able to handle it later. And think about your questions. So do you really need genomic data? What kind of data do you need? Or how much data do you think you're going to need? Okay, so molecular phylogenetics are essential tools. They're very helpful for plant taxonomy, and they go beyond what morphology can tell us. We know that morphology can be very deceiving because plants adapt to different environments. So things that look alike, not always closely related, and that can give us deceiving hypotheses. So molecular is great to help us with that. But integrating morphological information is still very important, so don't stop looking at the plants, please. Herbarium specimens are a very rich resource for molecular phylogenetic studies, so don't walk away from them. Keep collecting. If you don't use it for molecular studies, someone may do one day, so keep doing expeditions, keep doing taxonomic basic projects. Just take some cautions when sampling from herbarium specimens, just knowing what the issues could be. Today there are many different molecular methods, from very short genes to the whole genome. So just choose the laboratorial and computational methods that are most adequate to your research question, and we then the capacity of your lab group, of your institution, of your project, or even budget-wise. All of this needs to be considered. And talk. So like I've been doing for a long time so far. This is a field that's evolving very, very rapidly, so no one has definite answers for anything, like what you were saying. So what happens with herbarium specimens with next generation? We're still finding out, I think. So we're still discussing. So talk to people who know about different techniques, talk about colleagues who work on similar plants, and keep getting advice. Just don't jump into the project until you're well aware of what's going on afterwards. And that's it. Thank you for your time.